the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, here we are in our last session of Plant Kingdom, where we will be exercising both Gymnosperms and Angiosperms today. Angiosperms we will be learning in connection with sexual reproduction of flowering plants beta, and we will look into the alternation of generations. After that, life cycles, haplontic, haplodiplotic and diplotic life cycle, and then we will look into the previous year questions. Clear, right? Keep your mind here, okay, forget about everything, keep your mind here, with presence of mind listen to the class. After class we will discuss, okay, yeah. So yes, looking into the Gymnosperms, first let us look into the general characteristics. They are what? Embryophytic, tracheophytic, archegoniate phanerogams, okay, archegoniate flowering plants. Here the female sex organ is archegonia only. They are majorly known for primitive seed formation. They are majorly known for primitive seed formation. No problem. Call this name just. So here they are primitive. They are in the major characteristic of Gymnosperm is primitive seed formation, where seeds are naked without the seed coat. Okay, seeds are naked without seed coat. Fine. And what the seeds are without seed coat, because in pre-fertilization, that is before fertilization, ovules are not surrounded with ovary wall. Okay, ovules are not enclosed by ovary wall. We all know that ovule post fertilization will turn to seed. Ovary wall will mature to seed coat or fruit wall. Okay. So, before fertilization, ovules are not enclosed by ovary wall. So, post fertilization, seeds are not covered by seed coat or fruit wall. Next, plants are from medium size to tall trees and shrubs. Habitat is terrestrial, mostly mesophytes. Okay, and the smallest Gymnosperm is Zamia. Important to remember, beta. Smallest Gymnosperm is what? Zamia. Largest Gymnosperm is what? Sequoia. Okay. Living fossil, Gymnosperm which is living fossil is what? Ginkgo biloba. So these are few general characteristics. Major characteristic feature is what? Seeds without seed coat, which is primitive seed formation. Okay, and then roots. What will be the root system in the Gymnosperms? Root system is the usual taproot system, but in few of the Gymnosperms, for example in Pinus if you see, the roots are with fungal associations. Okay, roots are with fungal association, then that such root system in Pinus is called as what? Mycorrhiza. This is a symbiotic association, beta. So yes, taproot system is with what? Parent root and the lateral roots like this, right? So this parent root and lateral roots are associated with what? Mycorrhizal associations, okay, fungal associations. So fungal associations will increase the thread-like structures, hyphae, whereby the absorption capacity of water and minerals will be increased by the plant. Fungal association with the root system of Pinus will increase what? Absorption capacity of water and minerals. In turn, fungi is getting what? Shelter on the roots. Okay. So this is a symbiotic association. And then in Cycas, if you see, Cycas Gymnosperms, coralloid roots are observed. What are these coralloid roots? Is symbiotic association between the roots and blue-green algae. Blue-green algae, that is cyanobacteria, is well known for what, beta? Nitrogen fixation. Okay, so blue-green algae here, name few cyanobacteria or blue-green algae. Nostoc, Anabina, Oscillatoria, Alocera, all these are the blue-green algae. So what do? They aid in nitrogen fixation. In turn, what they are getting? Shelter on the roots. Okay, so this is the benefit of the symbiotic association with cyanobacteria, that is blue-green algae. This is what? Coralloid roots. Okay, mycorrhizal roots and coralloid roots, is that clear? Okay, mycorrhizal roots are observed in what? Pinus, coralloid roots in Cycas. This is the symbiotic association with fungi, and here this is the symbiotic association with blue-green algae. And here one peculiar feature in the roots is, usually in the taproot system you can see root hairs and root caps, but here there is no significant appearance of what? Root hairs and root caps. There is no root hair or root cap in this root systems. Clear na beta? Next is what? Stem. Okay, stem is unbranched in Cycas, branched in Pinus and Citrus. Okay, unbranched stem in Cycas, branched stem you can observe in Pinus and Citrus. Important liners from the NCERT. And then stems show U-stele. In Pteridophytes, which are the primitive vascular plants, if you have observed, we have observed that there you have primitive stele, that is protostele. But here in Gymnosperms, some advanced level of stele formation, that is what? U-stele. Okay. Next is vascular bundles. Vascular bundles are of what type beta? Conjoint, collateral and open type. Tell me what is conjoint type of vascular bundle? We have discussed in anatomy. I think not yet started for you, no? Yeah. Conjoint means nothing but, see, in a stele, what do you consider a stele as? Stele includes a pericycle, vascular bundles and pith. Stele includes what? Pericycle, vascular bundles and pith. Right, Nama? Okay. So, this is the pericycle. In that you have what? Vascular bundles like this. So, xylem and phloem, if they are at the same radius, there that is called as conjoint. Xylem and phloem vascular bundles are lateral to each other, which aid in the lateral conduction of water and minerals. This is called as what? Collateral. Conjoint, collateral. Clear, right? And open type of vascular bundles means there is a cambium present in between xylem and phloem, that is called as open type of vascular bundle. Clear with the terms conjoint, collateral and open? Clear, right? Next. So, vascular bundles, if you see in xylem, usually in the advanced plants like Angiosperms, xylem will have 4 tissues, phloem also will have 4 tissues. But here xylem, in xylem, vessels will be absent. What are those 4 tissues that you can see in Angiosperms? Tracheids, vessels, xylem fibers and xylem parenchyma. Amongst that, what are absent in Gymnosperms? Vessels are absent in xylem. In phloem, what are absent? Companion cells are absent. Companion cells are absent. In place of companion cells, what are present? Albuminous cells. And sieve tube cells are also absent. Good. Sieve tube cells are absent. They are present in the form of what? Sieve cells. Clear, right? One is xylem. In xylem, vessels are absent. Phloem, companion cells are absent. They are present in the form of albuminous cells. Sieve tube cells are absent. They are present in the form of sieve cells. Till here it is clear, right? Next, secondary growth occurs in the stems and roots because they have open type of vascular bundles. Cambium is present. We can observe both the cork cambium and vascular cambium in Gymnosperms, which will conduct the secondary growth both in stem and the root of Gymnosperms. Clear with the general characteristics, Noma? And leaves, if you see, pinnately compound leaves are seen in Cycas, okay, and needle like leaves which will prevent the process of transpiration, okay, that is observed in pinus plants like what? Conifers. Needle like leaves are observed which reduces the surface area, thereby prevents the process of transpiration. And then fan shaped leaves are observed like ginkgo, okay, in ginkgo, which is a living fossil of Gymnosperms, fan shaped leaves are observed. So, leaves adapted to withstand extreme temperatures, humidity, wind, thick cuticle, sunken stomata. This will help to reduce water loss. Okay, this will help to reduce what? Water loss. Clear with the leaf structures? This is all about the vegetative structures. So, in Pteridophytes you have observed for the very first time you have observed one feature, that is heterosporous condition. Most of the Pteridophytes are homosporous, few of the Pteridophytes are heterosporous. Name few Pteridophytes which show heterosporous condition. I have given you one mnemonic also. Chalo, open the running notes. Marsilia, Azolla, Selaginella, Salvinia. Okay, these are few Pteridophytes which will show what? What condition? Heterosporous condition. So despite that, which is the first evolutionary step in the pteridophytes, in gymnosperms you can see all the gymnosperms will show what? Heterosporous condition, wherein it will produce two types of spores, one is microspores and the other is megaspores. Microspores will develop into what? Male gametophyte. Okay, microspores develop into male gametophyte. Megaspores develop into what? Female gametophyte. Microspores landing on the suitable substratum, they will develop into male gametophyte. Megaspores develop into female gametophyte. So gametophytes, here in gymnosperms, the main plant body is sporophyte. Okay, main plant body is what? Sporophyte. So the root, stem, leaf, everything are diploid sporophytic body. And till pteridophytes you have observed beta, gametophyte is independent. Okay, gametophyte is independent. Yeah, but in gymnosperms, gametophyte is dependent on the sporophyte. Okay, gametophytic phase is dependent on what? Sporophyte. Okay, sporophytic plant body is independent from gymnosperms. So gymnosperms is having what life cycle? Diplontic life cycle. Gymnosperms are having diplontic life cycle. Next is what? Sporophylls. Okay, so on the leaf-like structures, if you are having strobili or cones, okay, over the leaf-like structures, sporangia are present, that is sporophyll. This feature we have observed in pteridophytes also. But in few pteridophytes like Selaginella and Equisetum, you have observed sporangia has compressed into strobili or cones, compact cone-like structures. Same ways in the gymnosperms, if you can see, sporophylls are spirally arranged along the axis. Okay, sporangia is compressed into cone-like structures, which is called as what? Strobili or cones. If megasporangiate is bearing on that, that is called as what? Female cone. Okay, if male gametophytes are there, like microspores are there, microsporangia is there, that is called as male cone. Female cone and male cone. Understood? Yeah. Here you see both sporophylls. If microspores are present, that is on those leaf structures which are spirally arranged over the axis, it is called as what? Microsporophylls and megasporophylls. Two different types of structures are observed. Microsporophylls, if you can see, they are compactly arranged to form what? Male strobili or microsporangiate or male cone. Male strobili, microsporangiate or male cone. So microsporangia, these are diploid, no beta? Okay. So microsporangia which are diploid, of which one cell you can name it as microspore mother cell, which is diploid. Okay. This microspore mother cell will undergo sporic meiosis. Sporic meiosis and form haploid microspores. Got it? Haploid microspores. This haploid microspores will develop into the male gametophytic structure, and the male gametophytic structure over here is pollen grain. Okay. So you have understood this clearly, right ma? Microsporophylls, the compactly arranged structure in this are what? Male strobili, microsporangiate or male cone. They will bear what? Microsporangia or microspore mother cell, which is diploid. This
difference between sporic meiosis and regular meiosis?
Nothing like. Here spore is undergoing meiosis, so it is called as sporic meiosis. No difference. The cell which is undergoing the meiosis, based on that the name is given. If you can consider the fungi and algae, thallophytes, their zygotic meiosis is happening, where that zygote which is diploid is undergoing meiosis. So you are calling it as zygotic meiosis. Here the spore is undergoing meiosis, so it is called as what beta? Sporic meiosis.
How many spores is it producing per mother cell?
One microspore mother cell undergoing sporic meiosis will produce 4 haploid microspores. Clear na? This 4 haploid microspores will mature or develop into male gametophyte, that is pollen grain.
Yes.
Clear, right ma? Yeah. So megasporophylls, here you see female strobili or megasporangiate or seed cone. These are the synonyms that you can give for the megasporophylls. What is it called as? Female strobili, megasporophyll or seed cone. Okay. So this has integuments over it. So unlike pteridophytes, if you see, there are no integuments present in the pteridophytes because of the absence of pteridophytes only. You can see precursor to seed habit, but there is no seed formation happening in the pteridophytes. Okay, you can see that female gametophyte is only restoring for variable periods of time because of the absence of integuments. But here integuments are present. Okay, integuments, inside that what? Archegonia are present, nucellus cells are present and micropylar end. Okay, once again I will describe you the structure of seed cone or female strobili. Female strobili is encoded with what? Integuments, and inside this seed cone you have what cells? Nucellus cells. Okay, few of the nucellus cells will be differentiated to what? Archegonia. Okay, there will be more than one archegonia usually. Okay, and over the one end, over the above end, you have what? Micropyle. Micropyle is the opening of the seed cone. Integuments, nucellus cells, one or few of the nucellus cells will turn into what? Archegonia, and the opening pore over this, over the apical region is what? Micropylar end. Clear till here, no? Now you see, one of the nucellus cell will turn to what? Megaspore mother cells. Megaspore mother cell will again undergo what meiosis? Sporic meiosis. Okay, and it forms how many megaspores? 4 megaspores. Of this 4 megaspores, 3 megaspores will be degenerated. Okay, and 1 megaspore will be functional. This functional megaspore will undergo sequential mitotic divisions. Sequential mitotic divisions. Okay, after sequential mitotic divisions, this will form multicellular female gametophyte which will bear 2 to 3 archegonia. Okay, so these are retained within the megasporangium, right? So they are undergoing mitosis and they are turning into many celled conditions. So those cells are called as endosperm over here. Here if you can see clearly, before fertilization only endosperm formation is taking place. Okay, so here endosperm formation is a pre-fertilization event, and here endosperm ploidy is what beta? Haploid. Endosperm ploidy is what? Haploid. Endosperm is forming pre-fertilization and its ploidy is haploid. Clear, right? Any doubt anywhere? Yeah. So here megaspore gives what? Endosperm, which is what? A pre-fertilization event which is haploid. Okay, yeah. So pollination. Now for the process of fertilization, first pollen grains have to reach out to what? Egg cell. Yes or no? So they will reach out to the egg cell through various agents of the pollination. The most widely used agent in the abiotic agents of pollination is anemophilus, that is wind pollination, through which pollen grains will reach what? Egg cell. Till pteridophytes you have learnt the pollination mode or the travel of the pollen grains as zooidogamous oogamy, where pollen grains will swim across the water and reach the egg cell. But here in the gymnosperm this is siphonogamous oogamy. Okay, because pollen grains are travelled through some tube like structure, that is pollen tube. Okay, siphonogamous oogamy. So post fertilization, the very first cell which is formed after fertilization or the product of fertilization is what? Zygote. Zygote is the product of fertilization. Zygote is diploid. Okay, this diploid zygote undergoes mitotic divisions and they will form diploid egg cell. Okay, and ovules will turn to seed, but there is no ovary wall, so seeds are naked without seed coat. Till here it is clear now? Any doubt anywhere? Yeah. Next is cycus. So here cycus is showing few of the fern characters, means few of the features which are in resemblance with pteridophytes. Okay, cycus shows both siphonogamous oogamy and zooidogamous oogamy. Okay, and male gametes in cycus are multiciliated. They are carried by what? Pollen tube. Fine. Fern characters which are seen in the cycus include circinate venation of young leaves, means the leaves will roll inwards like this towards inner side. This is called as circinate venation. This is one of the fern characteristics observed in what? Gymnosperm, that is cycus. And presence of ramenta. Ramenta are nothing but hair-like structures. There you have seen paraphysis and here it is ramenta, hair-like structures. Paraphysis, hair-like structures which are present over the Plant. Hair like structures which are present. Okay, that is ramenta. Next, presence of archegonia, that is a female reproductive structure. Archegonia is seen in pteridophytes and it is seen in cycas as well. Next, gymnosperms have three classes. What are they? Cycadopsida, Coniferopsida and Neetopsida. Cycadopsida best example is cycas, Coniferopsida example is pinus, Neetopsida example is neetum. Okay, Cycadopsida cycas, Coniferopsida pinus, Neetopsida neetum. Clear? Any doubt anywhere, ma? So here you look into the life cycle of gymnosperms once. First, a mature sporophyte. The main plant body is what? Sporophytic plant body. Okay. This sporophytic plant body will have male cone and female cone. Male cone called as what? Pollen cone, and female ovulate cone also, or seed cone also we can call it as. So this will bear what? Microsporangium. Sporic meiosis will form microspores that matures to pollen grains. And you can see wing shaped pollen grains in pinus. Wing shaped pollen grains in pinus, okay, which are haploid. Fine. And here ovulate cone or seed cone, again meiosis happens. Megaspore is formed which is haploid. One functional megaspore will mature forming a female gametophyte and fertilization will take place. Post fertilization, the first cell formed is what? Zygote. Develops to what? Embryo. Zygote is what? Diploid. Embryo is what? Diploid. Okay. And then seed is formed which is diploid. The seed will mature to what? A mature sporophyte. But seeds are without seed coat. That's it. Till here it is clear, no? No. Normal seed. Pollen grain is winged. No. Clear, no? Any doubt anywhere? Now about angiosperms. Gymnosperm story is clear, no? Yeah. Now angiosperms, you can describe angiosperm as embryophytic, non-archegoniate, vascular, fruit bearing, phanerogams or spermatophytes. Both gymnosperms and angiosperms are what? Seed bearing plants, spermatophytes. Smallest gymnosperm is what, na? Zamia. Smallest gymnosperm, Zamia. Smallest angiosperm is what? Yeah. Largest gymnosperm is what? Sequoia. Yeah, redwood tree. And largest angiosperm is eucalyptus. Clear, right? Any doubt anywhere? Yeah. And here, vascular bundles. Vascular bundles, xylem and phloem. Xylem includes 4 tissues in it. What are they? Xylem fibers, tracheids, vessels and xylem parenchyma. Xylem fibers, tracheids, vessels and xylem parenchyma. And phloem, if you see, phloem fibers, sieve tubes, companion cells, phloem parenchyma. Okay, phloem fibers, sieve tubes, companion cells and phloem parenchyma. Till here everything is clear, no? Next, reproductive organ. What is the reproductive organ in angiosperms? Flower. This is one of the characteristic features of the angiosperms. So in flower, if you see, beta, there are 2 essential walls and 2 non-essential walls, which you clearly learn in the sexual reproduction of flowering plants, 12th standard NCERT. Okay. So what are 2 essential walls? Essential walls and 2 non-essential walls. Two non-essential walls include sepals and petals. Petals and sepals. Okay, which is walls include calyx. No, over the thalamus all the essential and non-essential walls were placed. Thalamus is the base, beta, over which the essential and non-essential walls were placed. So here non-essential walls include calyx and corolla. Calyx have sepals. Corolla has what? Petals. Clear? So essential walls over here includes what? Gynoecium, androecium and gynoecium. Take down parallel. Okay. Gynoecium includes pistil. Okay, pistil can be monocarpellary or multicarpellary. Monocarpellary or multicarpellary. One carpel is a single unit of stigma, style and ovary. Stigma, style and ovary. Okay, stigma, style and ovary. Fine beta. Okay, one carpel is a single unit of stigma, style and ovary. Female reproductive organ is what? Gynoecium. Gynoecium is represented by pistil. Okay, pistil can be monocarpellary or multicarpellary. Monocarpellary or multicarpellary. Single carpel unit is what? Consists of stigma, style and ovary. Single stigma, style and ovary. Multicarpellary means many components of this. Androecium includes stamens. Single or many stamens. Each stamen will have what? Anther and filament. One long anther, bilobed anther and a long filament. Filament proximal end is attached to what? Thalamus. Thalamus. It is at the base of the thalamus, no? See, this is the filament if suppose.
Sometimes it's attached to the sepals and petals. Sometimes it's attached to the sepals.
No, its base is over the thalamus. Few times it is in the adhesion and cohesion. That concept will be learnt clearly in that chapter. Okay. I'll tell you, see. Stamens can be adjoined with sepals or petals or sometimes gynoecium also. And few of the stamens are arranged in the form of a bunch. Okay. That you will learn in the concept of cohesion and adhesion of the stamens. Okay, there you will learn it. So, that is adjoining or attachment, but at the base it is starting from where? Thalamus. It is adjoined to what? Thalamus, to stand on the thalamus. It is standing on the thalamus and it can adjoin around the stamens or it can adjoin with the other worlds, be it with gynoecium or with petals. Understood? Yeah. There we will learn it more clearly, ma. So, you are clear with essential and non-essential worlds, right? Any doubt anywhere here? Yeah. And then male gametophyte. Male gametophyte in the angiosperm is what? Pollen grains. Pollen grains are the male gametophytes. So, the pollen grain structure let me describe in brief here. It is not given in this chapter, but for your idea. Pollen grain will have an outer wall that is called as exine and inner wall is what? Intine. Mostly the pollen grain is dispersed in two-celled condition. One is vegetative cell and the other is generative cell. Okay. The cell wall, the exine wall is made up of sporopollenin, which is undestructible. Okay. Few regions of the exine are not with the sporopollenin depositions. From this region germ pore will be arising. Okay. Germ pore. Okay. From this region only pollen tube will grow when pollen grain lands on the stigma. Okay. And inner wall is what? Intine. Intine is made up of cellulose. Clear, right, ma? And the female gametophyte is embryo sac. Embryo sac is present inside the ovule. Okay. Ovule is present inside the ovary. To brief you in clear, I will give you a rough diagram. Stigma, style, ovary. Clear? Inside ovary you will have ovules. Okay. Let me draw one arrangement of ovules. You tell me what arrangement it is. The arrangement of ovules inside the ovary, in morphology you will learn. What is that? That concept is called as placentation. Arrangement of ovules inside the ovary. If they are arranged around the ventral suture like this straightly, it is what type of placentation? Marginal placentation. Inside ovule, ovule is diploid. Okay, remember that. Inside ovule, the cells which are present are called as nucellus. Okay, what are these cells called as? Nucellus cells. Nucellus cells are what? Diploid. One of the nucellus cell will differentiate into megaspore mother cell, which is diploid. After sporic meiosis, reductional division, they will form 4 haploid megaspores. 4 haploid megaspores in a linear fashion, of which 3 will degenerate, 1 will be functional. This will undergo 3 sequential mitotic divisions and they will form haploid embryo sac. I have briefed you the story, but this concept you will learn it more clearly under sexual reproduction of flowering plants under megasporogenesis. Here if you are clear with, there it will be easy for you to understand better. Clear, right? And here let us get into the structural description of the embryo sac. So, the embryo sac have 2 ends beta, one is chelazal end and the other is micropylar end. One is what end? Chelazal end and the other is micropylar end. Chelazal end will have 3 antipodals, okay. These are what? Haploid. Micropylar end have 2 synergids and 1 egg cell, 3 combinedly you will call it as egg apparatus. Egg apparatus includes 1 egg cell and 2 synergids, and at the center you will have polar nuclei, okay. Polar nuclei is also known as what? Central cell, okay. This is in n+n condition or diploid also you can say it as, okay. So, totally you can see 7-celled 8 nucleated condition, okay. How many celled ma? 7 celled. How many cell? 3 antipodals, 2 synergids, 3 plus 2 5 plus 6 egg cell plus 1 cell, that is central cell. Total 7 celled and 8 nuclei because central cell is having 2 nuclei. So, embryo sac is 7 celled 8 nucleated condition. Clear, right ma? So, here you see double fertilization. Double fertilization is nothing but which is a combination of triple fusion and syngamy. Okay, double fertilization means triple fusion and syngamy. Triple fusion means when pollen tube enters into the embryo sac, 2 male gametes will be released. One male gamete will fuse with female gamete, that is egg cell, that forms diploid zygote that develops into diploid embryo sac. Okay, this process of fusion of one male gamete with one egg cell is called as what? Syngamy. Another male gamete fuses with 2 polar nuclei. Here 1 nucleus of male gamete and 2 nuclei of the central cell will be fused. As 3 nuclei are involved in the fusion, you will call it as what? Triple fusion. And in the product, how many nucleated condition you can observe beta? Triploid condition, that is primary endospermic nucleus. That gets matured to triploid endosperm. Clear, right? Any doubt anywhere? Yeah. Now here the life cycle of angiosperms, and here angiospermic plant body is also sporophytic plant body. Only haploid cells here you see is male gamete and female gametophyte. Only male gametophyte and female gametophyte are haploid. Rest all the stages of the life cycle in angiosperms are diploid. So, what life cycle it will have? Diplontic life cycle. Here you see from mature sporophyte. Mature sporophyte is diploid. Young sporophyte, mature sporophyte, diploid. Seed is diploid, right? From seed develops into young sporophyte, mature sporophyte. Reproductive organ in this mature sporophyte is flower. Megaspore mother cell inside this. Just now I told you. Where do you see megaspore mother cell? Inside the ovule. Ovule. Ovule is present inside the ovary. So, this is diploid. Forms after meiosis, this forms 4 microspore tetrad. Fine. And here in pollen grain, that is in the male gametophyte, tube cell will be there, vegetative cell and generative cell. Okay. And finally, sperms means what? Male gametes will be released into the embryo sac. They will enter into the embryo sac. Diploid zygote is formed after fusion. Triploid embryo sac endosperm is formed after triple fusion. Diploid zygote after syngamy. This will mature into triploid endosperm. Diploid zygote will mature into embryo. Embryo turns into seed. Clear, no? So, complete all stages are diploid except male gametophyte and female gametophyte. So, the complete life cycle is what? Diplontic life cycle. Clear with the life cycle of angiosperms now?
Microspore mother cell arises from new cells also.
Microspore mother cell, that is a different concept where you will learn under microsporogenesis. We will learn it later. Okay. Angiosperms division has two classes, that is dicots and monocots. Few peculiar features where you can easily differentiate between dicots and monocots. The dicots have 2 cotyledons, monocots have 1 cotyledon. Okay, and venation, if you see, dicots have reticulate venation which is having branched veins, monocots have parallel venation which has veins like this arranged parallelly to each other. And flowers, whorls, I said, you know, petals and sepals, they are arranged 4 to 5 in the dicots, multiples of 3 you can see in the monocots. These are few characteristic differences. 4. Okay. And if you can see, root system is mostly taproot system in dicots, fibrous root system in monocots. And then plant life cycle, if you have observed all the life cycles from primitive plant division to the advanced plant division in the plant kingdom, where we have looked into what beta? Algae, Bryophytes, Pteridophytes, Gymnosperms and Angiosperms. Be it haplontic life cycle, haplodiplontic life cycle or diplontic life cycle. In any of the life cycle, alternation of generation is common. That diploid sporophytic phase is altering with the haploid gametophytic phase. This is alteration of generations. Okay. This is the observed in all the divisions, right? Yeah. So, here you see haplontic life cycle. Okay. First is haplontic life cycle. You have observed haplontic life cycle where in the algae, that is primitive division of the plant kingdom, wherein the gametophyte is what? Dominant, photosynthetic, free living gametophyte. Okay. Dominant, photosynthetic and free living gametophyte. Sporophyte is just single celled zygote. That is why, as there is only one stage, that is zygote, which is diploid, whereas all the stages of the life cycle are haploid. That is why it is what? Haplontic life cycle. And here what meiosis will happen? Zygotic meiosis. Examples under this you can take it as volvox, Pyrogyra and some species of Chlamydomonas. Okay. But here from the algae also there are few exceptions. I have told you when I am explaining you the algae, like few of them will have haplodiplontic life cycle like Ectocarpus, like few of them will have diplontic life cycle like Fucus. I have told you under exceptions, I do not know how far you have remembered it, just revise it once again, you will get into it. Haplodiplontic life cycle where gametophyte is dominant in Bryophytes, gametophyte is independent of the sporophyte, sporophyte is dependent on the gametophyte. This we have learnt in the Bryophytes. Okay. Next is dominant sporophyte. You can observe this is in what? Pteridophytes. In Pteridophytes, what is the dominant phase? Sporophytic phase is the dominant one. And here sporophyte is independent of the gametophyte. Sporophyte is independent of gametophyte. Gametophyte and sporophyte both are independent to each other, but which is dominant phase? Sporophytic phase. Okay. Yes, in Pteridophytes. Dependent sporophyte you can see in Bryophytes, independent you can see it in Pteridophytes. Some algae, I said you know there are exceptions which has haplodiplontic life cycle. Some algae like Ectocarpus, Polysiphonia and kelps will have what? Haplodiplontic life cycle. Clear right Nama? Any doubt anywhere? Examples are important, exceptions are most important. Diplontic life cycle, first is sporophyte. Sporophyte dominant stages will be present, only gametes or gametophytes which are haploid, only those stages are haploid, rest everything is diplontic phase. Sporophyte is dominant, photosynthetic and independent. Okay. Gametophytic phase is fused celled, haploid gametes. So, all seed bearing plants, these are all what? Seed bearing plants. For example, Gymnosperms and Angiosperms. Okay. Fucus is the brown algae which has what? Diplontic life cycle. Okay. Now, few questions, NEET PYQs. First question Raghav, do it. Good evening Sachita, I hope you have gone through the session. Work out the objective beta. You can drop your answers ma. And if you are having any doubt, feel free to ask.
Fourth option.
Sure? Which one of the following is characteristic feature of gymnosperms? Primitive seed formation, that is seeds are neat. Good. Second question.
Third option.
Carefully see and answer, that is on the examples.
Says, ma'am, Sequoia comes under Angiosperm.
What is the tallest gymnosperm, Raghav? Edi.
First option.
Just a second, ma. Just a sec. Yeah. Abdul, good. Second question, which option?
First option.
Good. Gymnosperms include Citrus, Pinus and Sequoia. Angiosperm doesn't include Sequoia. This comes under gymnosperms. Bryophytes, Polytrichum, Polysiphonia and Sphagnum. No. Sphagnum? No. Polysiphonia is the algae. Which algae? Red algae. Pteridophytes, Equisitum, Gingo, Adiantum. Gingo is not a pteridophyte. It is a living fossil, comes under gymnosperms, right? This question. Assertion reason asked in NEET 2023. Do it, ma. Is it? In gymnosperms, the pollen grains are released microsporangium and released from microsporangium and carried by air currents. True? Anemophilus. Air currents carry the pollen grains to the mouth of the archegonia, where the male gametes are discharged and the pollen tube is not formed. Pollen tube. Yes, you are right. So here what is the option? 1. Okay. Next question. Last question of today's session, mark it properly. Take your time and do. Online students, even you can answer, ma. First tell me, Spirogyra is what? Comes under which division of plant kingdom? Algae. Fawn. Pteridophyte. Funaria. Cycas. Good. Now you answer. Decode the question first. If you decode the question, it will be easy for you to answer. Okay, let me check it out. Yeah. So Spirogyra, dominant haploid free living gametophyte. Okay, accepted. B is 4, you said, right? Fawn, dominant haploid leafy gametophyte altering with. In pteridophytes, what is the dominant phase, beta? Sporophyte, I told you, no? Dominant diploid sporophyte vascular plant with highly reduced male or female gametophyte. No. B will be 3 because it has dominant diploid sporophyte alternating with reduced gametophyte called as what? Prothali. B is 3. A is 2, A2, B3. C4, Funaria, dominant haploid leafy gametophyte alternating with partially dependent sporophyte. What happened to you? D is 1, Cycas, which is gymnosperm, dominant diploid sporophyte vascular plant with highly reduced male or female gametophyte. Clear? So, what is the right answer? Option 1. Good, Abdul. Okay. So, that's all with today's session today. So, here we have completed what? Plant kingdom is done, and the next chapter is morphology in higher plants. We will look into that in the next session. That's all for today. Thank you.